bioinformatics analysis. So this is genomic DNA isolation. I do not need to go into the details. So just one thing, for NGS we have to quantify the DNA using the qubit fluorometry. We do not rely on spectrophotometer technique like Nano-pore. We have to use fluorometer for the quantification of exact amount of DNA. And for the purpose of DNA purity, we can check Nano-pore 260 by 280 ratio, which must be more than 1.7. Even if we get 1.5, 1.6, the sample can work. So this is the first step in library preparation. So this is just the theoretical aspects how library is prepared. I presume that some of or many of you already know how the process works. So what was happening in Sanger sequencing, we just amplified only one portion of DNA into millions of copies. So those fragments were equal in length and the sequence was same. But here, with the genomic DNA, we do not amplify. We just break the whole genomic DNA into millions of fragments. So one thing is that these fragments are not equal in length, they are different in length because this breakage of DNA is random. This does not follow a principle, a defined length. This is random, and the output DNA are of different length. The second thing is that the sequence of this fragment is different from each other because these fragments represent the whole genomic DNA, not a single targeted region. So how this is accomplished? There are different ways. One is enzymatic and the other is mechanical way. So what is the mechanical way? Using sound waves. So there are different instruments like sonicators or covares which produce a sound wave of specific intensity. Those waves strike the genomic DNA and breaks the DNA into different fragments. So this is one method. The other method is enzymatic. So there are certain enzymes like nucleases. Those enzymes cleave the double-stranded DNA at different places randomly. There are also transposases. Some kits use nucleases and some kits use transposases. Transposases also use a double cut in the DNA fragment. So using any technique, we break the whole genomic DNA into millions of fragments. So these fragments are of different length and of different sequencing, which represent the whole genomic DNA. In next step, so here I have a small question. This is our genomic DNA, which have been broken into small parts. So if I have to amplify all the fragments and I do not know the sequence of these fragments, how can it be possible to amplify all the fragments? In PCR, we use specific primers which target to a specific region. The sequence of those primers is very known to us, and then the target region is amplified. But here, the sequence of these fragments is not known. We have to sequence this DNA in the machine. So how is it possible to amplify all these regions? So scientists use the ligase enzyme. So there are specific oligos, double-stranded oligos, which are ligated to the ends of these fragments. The sequence of these oligos is known to us. So this step is called adaptase ligation. So these oligos are termed as adapters in the technology of next generation sequencing. So these adapters are ligated to the ends of DNA so that the library which was in this form comes to this format. So on each strand there are known adapters at left hand side and right hand side. The sequence of these adapters is known to us. So now we can use primers which are complementary to these regions, and those primers can amplify all these fragments simultaneously.
Can you call these adapters as indexes?
Yes.
Because in Illumina we use the plates. named indexes?
So indexes are advanced form of adapters. So another way is like in Illumina there are transposases, as I just told you that there are different ways. In Illumina they use transposases. So what is here? This is a two-step reaction. In first step, the genomic DNA is fragmented into hundreds of millions of fragments, and in next step, adapters are ligated to the ends of fragments. So this is a two-step process. So in Illumina, this step takes place in one. These two reactions takes place one step. In the same step, the transposases break the DNA and also attaches the adapters to the ends of both strands which have been newly generated. So as a result, the fragments are generated in this format. So this process is called fragmentation. This is acronym of fragmentation plus addition of tags in a single step. So now the genomic which was in this format has come to this format after the initial step of library preparation. So this is the simplest way of preparing the library for performing NGS sequence. There are also many other steps. If we have to pool the samples in one reaction, then we have to add indexes to these fragmented DNA so that we can pool together. So how we can add indexes? So now this is our fragmented DNA. I am just showing one fragment here. This is our unknown DNA, and these are the adapters which we have ligated to the ends of DNA. Now we have indexes, which I told you earlier. This is an advanced form of these adapters. So now these indexes, there are three parts of these indexes. This is approximately 45 nucleotide long oligos. The first portion is complementary to this adapter, both sides. The second part of this index is a unique nucleotide sequence which is approximately 8 to 10 nucleotides long. And the third portion is P7 at one side and P5 at the other side. So there are three portions of these indexes. So after using these indexes, we amplify these unknown fragments. So at the end of amplification, the fragments come into this format. So this is the adapter sequences which we added at the very earlier. This is the index sequences, and this is P5 and this is P7. So all the fragmented DNA now come to in this format.
सर इसी को ही हम ए टेलिंग टी टेलिंग और बारकोडिंग वाला स्टेप इसको कहेंगे कि कुछ वो डिफरेंट है सेटअप?
ए टेलिंग यहाँ पर है. दिस इज द ए टेलिंग स्टेप. So this is the fragmented DNA, right? We have to join the adapters at both the ends.
Right.
So for joining the adapters, we have to do
prepare the ends.
Doing A टेलिंग, we can just, this is the detail of how this step works.